So in this workshop, I'm going to talk about an introduction to genetics and monohybrid crosses. And so first off, we need to get a basis of vocabulary. So when we use this vocabulary, we understand kind of where everything is coming from. So let's first start off with a gene. So last semester we talked about DNA, not last semester, I guess it was last project, we talked about DNA and transcription and translation and how those genes are used to translate into proteins. And we talked about protein synthesis. So you should remember that a gene is simply a piece of DNA that codes for information within a cell. And so these genes are actually what code for specific traits that are exhibited within an organism. So your eye color, your hair color, etc. So then we have an allele, which is an alternative form of a gene. So I know you've had background in this, so whether that would be tall versus short, round versus wrinkled in pea plants, for instance, things like that. Now, with those alleles, you have different combinations. It could be homozygous, which would be two copies of the same allele at the same locus, which, if you wondering what a locus is, the locus is a specific location on a chromosome. Now, when they're homozygous, they're the same allele, so you could have homozygous dominant or homozygous recessive. So homozygous dominant would have two big letters like this. We always represent dominance by a capital letter, and we exhibit recessiveness by a lowercase letter. So most of you have probably heard of that before. So homozygous, they would have the same letter for both allele. Now, conversely, we have heterozygous, which would be two different copies of an allele at the same locus. So heterozygous, meaning if we were talking about letters in capital or lowercase, you would have one capital letter and one lowercase letter. In this instance, big H, little h. And these two things are sometimes confused, but you need to make sure that you can make the distinction between these as well. You have a genotype and a phenotype. So the genotype is the genetic makeup of an organism. So those are the actual letters that we represent when we do a Punnett square. And the genotypes can be homozygous, heterozygous, and it represents what the actual genetic makeup of that organism is. And then finally we have phenotype, which is the actual physical traits or characteristics that the organism exhibits. So it could be a round seed versus a wrinkled seed. Some of you have brown hair, some of you have blonde hair. Some of you have brown eyes, green eyes, blue eyes, etc. So it's whatever trait we're talking about, what that physical characteristic is when it's expressed in the organism. So the next thing we need to talk about is being dominant versus recessive. So a lot of times in middle school you probably learned it as dominant is the big letter and recessive is the little letter. So it's a little bit more than that. The dominance means that it's going to mask the recessive allele, and meaning that it's going to actually cover that recessive allele up when it's expressed. And you only need one dominant allele to be able to express that trait. So if we had like this heterozygous individual over here, big H, little h, you would see that you only need one of these here present to be able to express that specific trait. And then recessive, you would need two copies of the recessive alleles to for it to be expressed. So over here, when we talked about homozygous recessive, it has to have two copies of that allele present to be able to be expressed in the organism. And so that hopefully clears up the difference between dominance and recessiveness, and it's not just one letter's big and one letter's small. So if we move on now to using Punnett squares, I'm sure a lot of you have become pros at that, but we're going to take it a little bit further than just filling out the boxes. We're going to actually understand what those boxes mean. So when you fill out a Punnett square, you want to make sure that first you put the parents' alleles along the top and then along the side. And that's what those are actually expressing. You're showing the combination of alleles that parent can pass on to their offspring. And so you get one allele from each parent, and that's what makes a trait. So you have to have 2 alleles for a trait, but you need 1 from each parent. And so you're showing what their genotype is and what those possible alleles are that they could pass to their offspring. And then these boxes here are showing the 4 possibilities that could be passed on to their offspring. So it's kind of all probability. It's, one box is 25%. So you add up each of those 25% and you get the total of 100% and all the possibilities of what could be passed from parent to offspring. And so if we usually you put the mother across the top and you put the father down the side, and it just depends on what their specific genotype is. And so for instance, let's do an example. If we're talking about pea pods and we talk about round versus wrinkled, that's one that Mendel studied extensively. So if we talk about that, we're going to use round is going to be dominant to wrinkled. So we will say round is big R and wrinkled is little r. Okay, so these would be the constraints of this, and you always want to make sure that you're accounting for what is dominant and what is recessive. Now sometimes we won't tell you what the letters are, so you're going to have to come up with those on your own, but most of the time we'll give them to you so you can figure that out. So if we use an example where we are crossing 2 heterozygous pea plants. Okay, so first you'll need to figure out what their genotype is. So if both plants are heterozygous, they would both be big R, little r. So we always use an X in between to show the cross between the two. So this would be the genotype of both parents. Now, this is showing that this parent could give their offspring either a big R or a little r, and this parent could give their offspring a big R or a little r as well. So you just have to take that into consideration and put that on your Punnett square. So we would put a big R here and a little r here, and a big R here and a little r here. Now, you're going to combine those alleles in each box, and then that will show you the possible genotypes of the offspring. So here we'd have